Hello, and thank you for coming to this talk. My name is Adam Cooper. I'm a PhD student in atmospheric chemistry at the University of California, San Diego, where I research the atmospheric fate of organic UV filters as a part of the SLADE research group. Today, I'll be presenting on a collaborative project with Chemists Without Borders and the GOES Foundation, the Global Oceanic Environmental Survey Foundation, with major contributions from Kara Wiggin and Mickey Rogers. We are a team made up of academic, nonprofit, and governmental professionals around the world who have joined forces to use our resources and expertise to take on oceanic pollution. At Chemists Without Borders, we realize that chemists are responsible both for building the molecules that make our lives better and also their unintended impacts on the environment and human health. We have launched this project in collaboration with the GOES Foundation, which focuses on a very specific problem caused by pollution: synergistic toxicity of UV filters and microplastics on marine life. The combination of UV filter pollution and microplastic pollution makes a toxic contaminant cocktail that has impacts greater than the sum of its parts. This will have an effect on the entire marine ecosystem, but the GOES Foundation has remained focused on the planktonic species that are responsible for sequestering massive amounts of carbon dioxide, make up the base of the food chain, and ensure that the marine ecosystem can thrive. As I mentioned, phytoplankton are a tremendously useful ecological group that sequesters carbon dioxide from the atmosphere and produces the majority of the world's oxygen. This serves as a buffer for rising CO2 levels during the climate crisis, and the strength of this system has shielded us from experiencing the full warming potential of our emissions. However, these phytoplankton populations have been decreasing at an alarming rate of 1% per year, and we have already seen marine net productivity halve since 1950. While most scientists attribute dying populations to a change in climate, leading to ocean acidification and stratification, the growing polluted chemosphere and plastosphere in the ocean cannot be helping. Combining the levels that we see in environmental samples with toxicities we observe in experimental studies, there is a major emerging issue that does not seem to be slowing down. Losing the health of the oceans means losing the fight against climate change. If we can't save the plankton, it brings us one step closer to reaching a stable climate system and saving the planet. The first major class of pollutants we focus on are organic UV filters found in sunscreens and used as plastic additives. I show three here, known as the 3 O's: octinoxate, oxybenzone, and octocrylene. These present ecotoxicological effects, most famously oxybenzone causing the bleaching of coral reefs, but also on marine life from algae up to fish. They can biomagnify up the food chain, which presents another route of exposure to humans beyond their application as sunscreen agents. Oxybenzone specifically is tracked in my home state of California in a biomonitoring program for its impact on endocrine disruption as a phenolic estrogen mimic. These compounds also show genotoxic and carcinogenic effects. The second half of the title and of our project is focused on microplastics. The plastic industry is a dominant force in the Anthropocene and the theme of this ACS meeting. These materials are made from petrochemical feedstocks, also responsible for our climate crisis from the fossil fuel industry. While we reduce emissions, the petrochemical industry is shifting to continue the exponential increase in global plastic production shown on the left. However, a reliance on polymeric plastics and mishandling of the waste shown in the middle map has resulted in the creation of what is known as the plastosphere, an entire new environmental system made up of the building blocks of plastics. Large plastics degrade physically into smaller and smaller pieces known as microplastics, which range from 1 micron up to 5 millimeters, and nanoplastics, which are less than 1 micrometer. Some of the dominant primary sources include runoff from transportation, shedding from synthetic garments through the wastewater stream, and direct pollution from the global maritime industry. Secondary sources involve degradation from plastic litter in the oceans, which represents a major contribution. By 2050, it is expected there will be more plastic in the ocean than fish by mass. These particles float and accumulate in the sea surface microlayer, participating in sea-air exchange. They are also biomagnified from plankton to larger and larger fish, shown in the figure on the right, where many then transfer from the marine biosphere into humans. About 10% of the world's population relies on the ocean as a source of protein and employment, 95% in developing countries of that 10%. In all cases, the ocean does not act as a sink for this pollution. It stays close to and in our air that we breathe and the food that we eat. Here is a set of 2 maps showing real environmental concentrations of these pollutants in the oceans. UV filters are found most commonly roughly in the 100 ppt to 1 ppb range, making them major xenobiotic signatures in many environments, especially near wastewater treatment plants and recreational beaches. These beaches can see huge pollution spikes on crowded days, where concentrations can reach upwards of 1 ppm. As a marine atmospheric chemist, this is mind-blowing. The average concentration of marine dissolved organic matter is 1 ppm. In these areas, UV filters are the same concentration. They're completely dominating and changing the entire ocean-air interface. Microplastics have less standardized reporting parameters, and concentrations can vary greatly. Depending on the size ranges, different techniques can get down to. Generally, they are detected in the thousands of particles per cubic meter, but can be a lot more common when you have better techniques to study smaller and smaller plastics. Both classes of pollutants are nearly ubiquitous in the environment. Taken on their own, both UV filters and microplastics warrant their own remediation processes. However, there's a growing consensus that microplastics can compound organic pollution in the marine environment. Many organic pollutants of interest are hydrophobic molecules. This hydrophobicity means that they will preferentially accumulate on the surface of hydrophobic microplastic particles. Now, organic pollutants that were distributed in the marine dissolved organic matter pool are attached to a unique form of particulate matter that directly interacts with marine biota. Studies have shown that this accumulation can be upwards of 1,000 up to 1,000,000 times more concentrated on microplastics compared to their concentrations in seawater. Zooplankton graze on particulate matter and mistake microplastics for food. Microplastics can absorb onto the outside of phytoplankton, and nanoplastics may pass through their cell walls. In both cases, the microplastic acts as a potent vector for concentrating and delivering organic pollutants into marine life. I would like to highlight this study by Knott et al., where they observed this synergistic toxicity of microplastic fragments and BP3 or oxybenzone additives on a grazing zooplankton, Daphnia magna. It is important to note here that this is a relatively large freshwater zooplankton. There is yet to be a similar study on marine plankton, although this is a common proxy organism. They found having both compounds present led to an effective toxic dose, nearly an order of magnitude more dilute than when either was present alone. In the left plot, they found the presence of microplastics greatly contributed to the accumulation of oxybenzone inside this model species after 48 hours of exposure. This effect compounds with rising levels of pollution, shown by this huge jump at 0.1 mg per liter, or about 100 ppb. In the right plot, they also measured oxidative stress, which is indicative of sublethal toxicity. And each of these series on the left, you see reactive oxygen species formed from exposure to microplastics, on the right from exposure to oxybenzone, and in the middle upon exposure to both. You begin to see additive toxicity almost immediately and synergistic toxicity upon exposures to higher amounts of pollution. This synergistic toxicity is being observed in many macroscopic creatures, but is yet to be characterized on the base of the food chain, phytoplankton. Microplastics can adsorb onto the surface of these plankton, and nanoplastics may pass directly through their cell walls, potentially causing similar synergistic effects others have seen. We are pursuing these studies from both top-down and bottom-up approaches. The GOES Foundation is seeking to mobilize the recreational sailing community to participate in citizen science through their collaborative data collection project. They are forming partnerships with large recreational sailing organizations to promote citizen science data collection to make a global map of phytoplankton and microplastic counts. Using their Jesus filters and portable microscopes, sailors will investigate particulate material and count zooplankton, phytoplankton, and microplastics. They aim to collect about 2,000 samples in the next year and build a machine learning powered tool to map and interpret this data to observe real trends in phytoplankton populations and microplastic pollution. From the bottom up, we are also pursuing several collaborative projects through Chemists Without Borders to determine the full extent of this issue on the molecular scale. At the Scripps Institution of Oceanography, we are designing a synergistic toxicity assay with representative marine plankton and representative microplastic fragments to answer the lingering question in our project. Is the synergistic toxicity experienced by other model organisms also seen in marine plankton like we expect? My own thesis work, as a part of my PhD in atmospheric chemistry, is to study the air-sea transfer of pollution and the atmospheric lifetime of these emerging pollutants. Understanding their impact on coastal air quality can help motivate regulation that may not be as similarly motivated by environmental damage, given the priorities of many federal agencies and politicians. Additionally, we are working with a faculty member at the Federal University of Sao Carlos to better understand how polymers degrade in a marine matrix and how polymer chemists can build better materials which do not physically degrade as readily into microplastics. The last project I'd like to mention is our effort to detect UV filters and microplastics in the most remote stretches of the oceans, Antarctica. One of our team members will collect ocean water samples for dissolved organic matter analysis off the coast of Peter I Island this winter inside the Antarctic Circle. I will run these samples using a non-targeted LC-MS-MS workflow designed to detect xenobiotic signatures in dissolved organic matter to determine environmental concentrations of these compounds, some for the first time, depending on their presence above our limit of detection. Additionally, we are in the early stages of a larger agreement with a recreational cruise company to integrate water and air quality monitoring aboard their vessel, collect samples along their tracks, and perform public education and outreach with their guests. Finally, we are working to synthesize our background research and ongoing work into digestible content for policymakers and elected officials through the construction of science policy journal articles and concise policy memos. Here we have our main policy recommendations for managing UV filter and microplastic pollution. We support regulatory bans for the 3 O's, as seen in communities like Hawaii, Key West, Aruba, among others. In order for these bans to be popular, we need to expedite innovation through measures like the Sunscreen Innovation Act, which was introduced in 2014 and subsequently underfunded and then overturned during the last US federal administration. In fact, it was removed during the initial coronavirus relief bill, the CARES Act, which shows the danger of anti-science legislation sneaking into unrelated bills. Finally, we wish to spread awareness on UV filters and other endocrine disrupting compounds, similarly to California's Prop 65 for carcinogens and birth defect causing compounds. For microplastics, we aim to cut off pollution at the source through better resilience along the waste stream and investment into new technologies. For microfibers, this means introducing microfiber filters to washing machines to defend against synthetic fiber clothing shedding. For environmental runoff, this means the introduction of fine filters and other microplastic catchment technologies in stormwater and wastewater systems. And for the chemical industry, we need a robust investment in the creation of more resilient polymers while seriously cutting down on the mass of plastics created. We are a growing team and we thrive on collaboration with the experts such as you are, which is why we are here at ACS sharing our plan of action. As our president Ray likes to say, we have a platoon of volunteers. We need an army. If you'd like to join our army, please reach out through my email listed below. Thank you, and I'm happy to take any questions or have a brief discussion.